Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
My apologies for not showing up earlier in the meeting, but I had to teach my evolutionary genetics class this morning and then come here after that. And I'll be here until mid-afternoon, and then I have two more things happening today. But I'll be back tomorrow, and I hope to be here for the whole thing tomorrow. So if anybody needs to catch me about anything and you don't get me today, I'll be around tomorrow. I was asked to give a general introductory talk about phylogenies, and I think that probably would have been most useful at the beginning of the meeting instead of a little bit into the meeting. But I'll do it anyway, and I hope it's perhaps everybody here. I see a lot of people that I know this stuff, and perhaps everybody here will consider this routine review, but I think it's necessary for some of the people, so I'll just go ahead with this. And what I wanted to do was just go over some of the major approaches to inferring phylogenies, give some intuitive discussion of how they work, not a rigorous mathematical discussion. So I won't be proving theorems here. I'll just be giving you a feel for how these methods work. And then also talking about two other topics. And I was at a meeting once in which there was a lot of algorithmic discussion of how do you infer phylogenies and new algorithms for inferring phylogenies. And I said to the organizers of the meeting really at that point that was a mistake, because yes, that's important, but it's only about 1/3 of what you need to talk about. That about a third of it is how do you find phylogenies. But another third of it is, well, once you've found them, how much confidence can you have in different parts of that tree? Which parts are best supported? How much support is there for parts of those trees? The statistical question of what set of phylogenies do we have support for as opposed to other phylogenies? And that wasn't, at that meeting, that wasn't being discussed at all. In fact, they had one talk on it, and that was me. I was assigned to do that. So I was assigned to cover 1/3 of everything that they were speaking about. Then there was another third that nobody was covering at that meeting. And that is, well, what do you do with them once you get them? And for that matter, why are we bothering to get them? And I hope to talk about all three of these at least a little in the talk today, to try to give people a picture of the whole area of inferences involving phylogenies. That's right. Well, let me put it more accurately. When I sat down to do the talk, I said, maybe these people think it's a small topic that can be done in 1 hour. But this, in fact, I had just finished writing a few years ago a 580-page book on this topic. And then I give a course at the University of Washington every other year, and that's 30 hours of lecture, and it's absurdly short. We just skim through whole areas and leave whole topics out. And so I thought, well, now they've asked me to take those 30 hours, that inadequate 30 hours, and shoehorn it into one 50-minute block. So that's what you'll get. Whether it's what you deserve, I don't know. So this is what I'm really saying here. What are some of the strengths and weaknesses of different ways of reconstructing phylogenies? How can we find out how accurate we've been in reconstructing the phylogeny? And why do we want to reconstruct it, and what do you use them for? And I want to start by giving you a picture, much less sophisticated than the picture that I gathered Jun-Young Kim gave you earlier today, but I hope consistent with it, of what the tree space looks like, at least in a naive consideration. I'm sure it can be transformed into isomorphism with much more incomprehensible things, but I want to give you a simple case. So let's start with a really, really, really simple case. We have 3 species, A, B, and C, and we have a tree, and we're going to restrict the trees we look at by assuming a molecular clock. So we're going to assume that the expected amount of evolution up each lineage from the root of the tree is equal. That's a somewhat arbitrary assumption, but has some realism in some context. In that case, they're really, for bifurcating trees, there are only 3 of them, and it just depends on which 2 of these 3 are the sister species, AB or AC or BC. And there also can be a 3-way split with ABC. And they are parameterized by how far down in branch length you go till you get to this point, and how far down you go till you get to that. So it's 2 parameters. So that means you can plot those on a graph, and you'll find that T2 has to be bigger than T1. So you're in this triangle here, although the triangle goes off to infinity to the right. So it's an infinitely big triangular fin. And you then have to consider what happens as you move around on this fin. If you move down to here, T1 is 0, so these 2 are just forking at the last minute, and T2 is down here somewhere. If you move up on the fin, as you get up here, T1 becomes equal to T2. And in a case like that, the tree is in effect a 3-way split. If you have a model of evolution in which the fork itself doesn't cause any change, it's just evolving on the branch. There are models of evolution, punctuated equilibrium models, for example, where going through a speciation does something, and that would not have this property. But we'll leave that aside. As T1 gets equal to T2, it becomes equivalent. The data you predict arising on that tree is the same as it would be for a 3-way split. So, and if you have a different tree topology, say B going with C, as you move that coordinate down, you also come to the same tree, a 3-way split. So the 3-way split is intermediate between the 3 phylogenies. And so along that seam, you reach the edge of the 2 other possible triangular fins corresponding to the 2 other bifurcating tree topologies. And when you glue them together, here's the backbone. You get a 3-finned paper airplane that's going, but it's an infinitely long paper airplane, and that's what the space looks like. So it's got these real pieces, and they're glued together in a funny symmetrical relationship. I used to, in some talks, have a paper airplane with little trees drawn on it, and would imagine myself sailing it out over the audience. But we got worried about liability. And when you get to larger trees than 3 species, and you also relax the restriction of a molecular clock, for many of the models, many of the methods of inference that I'll talk about, the tree, it becomes unrooted. That is, it turns out that where the root is on the tree doesn't matter to the distribution of data predicted by that tree. That's true for many of the criteria that people use. So here's an unrooted tree. It has branch lengths. I'm just giving them this, calling them V and giving them subscripts. And we can imagine taking a tree like this and shrinking one of the branches, V9. And as you shrink V9, those 2 nodes come together. And when they hit each other, it's a 4-way split there. So you could imagine yourself being in a space which has a 9-dimensional space indexed by these V's. And this is a real 9-dimensional space. Well, I'm drawing it as a 3-dimensional space. And as we shrink one of these guys, we're moving along towards a wall of the space. And we can't get past the wall because negative branch lengths don't mean anything. They're meaningless. And so what happens as these 2 nodes hit each other, you come to a 4-way split. You can re-resolve it in 2 other ways. There are 2 other tree topologies there, depending on which partition of 4 things into 2 groups of 2 you make. One of them is this one, and then there's 2 others. So it's as if you're walking along and you're walking into the wall, and as you hit the wall, it's through the looking glass. That is, you arrive at the wall, and it's a magical place from which you can see 2 other rooms. And you have to choose which room you're going to step into. And then as you step into it, you're in another tree topology, and you're resolving, and then that branch length is getting bigger as you walk away from the wall. Now the question comes, okay, that's locally what it looks like. And then there are floors which would correspond to shrinking a terminal branch and an external branch, and that doesn't have anything When it hits 0, you don't go anywhere. So that you go down and hit the floor, you can't go through the looking glass at the floor, but you can on the walls. Now, how are these things arranged? How are these series of rooms arranged? Well, here's a case. This just shows that process. I'm sorry. It shows us shrinking V9 down to a point, and then you can go over to either this one or that one. They're the other two tree topologies you get for that exact same tree. I should have shown you that. And I once made up a graph of this, and I drew 15 little trees, and I drew lines between them, and it was a complete mess, and I handed it to my stepson, Ben Schoenberg, who did a master's degree in combinatorial geometry. And I said, Ben, arrange this graph for me. And this is what he came up with. So I call it the Schoenberg graph. It is very interesting. It's very high dimensional. It's very non-planar. Here are all the 5 species, the 15 5-species unrooted trees. And we've connected ones that can be reached from each other by this process of shrinking a branch to 0 and walking into one of the two alternative realities on the other side of the wall. And each time you do that, there's two different, it's hard to see, but there's 2 different internal branches here. One pair leads you this way to one of these 2, and the other pair leads you either this way or that way. And the 3 trees that are connected, they form a little triangle. There's a whole bunch of little triangles there. Yeah?
So this is sort of a nice mathematical model sort of thing, a visualization or whatever of this space.
Yeah.
But is there any biological reality through these walls? I mean,
Sure. The biological reality is that it's a tree in which there are 3 species coming from one species, because each lineage on that tree is a species evolving through time. And it comes to a point where a speciation event occurs. One species separates into 2 species, or into 3. And if it separates into 3, that's when you're at the wall.
But do you really have this in biology that brings this and change the tree? Or is it just
We don't change the tree because the tree of life was evolved out there long before we were born. And where it's not up to us to change it any more than we can rearrange galaxies. You know.
it's just a mathematical
Yeah, I could make a mathematical. Yeah, I could make a mathematical model of galaxies, and I could rearrange that model, but that won't shove a single star around. However, and the same thing is true here. What we're interested in is finding out where in this space the true tree is, basically. So in doing so, we have to move around the space and look at different things. So anyway, this graph is nice. Now then, the next one up with 6 tips is 105. It's 105 points. By the way, this has some interesting aspects, one of which is that these triangles share trees between them. You can make a dual of this graph by putting a point for each triangle and connect those by a line if they share a tree. And if you do that, you arrive at something called the Peterson graph, which is a famous graph in graph theory to the point where one mathematical journal has it on the cover. So the dual of this thing is the Peterson graph, which Ben figured that out. There's been some other tree topology people who've worked on trees. They came to the same exact realization independently. Ben's work was not published, but it was on a T-shirt at the Woods Hole Workshop of Molecular Evolution. Okay. How many trees are there? Well, you can work that out rather easily. This is work goes back more than almost 150 years. For unrooted bifurcating trees where each node has 3 neighbors, you can easily show that the number of topologies of them, ignoring the sizes of the branch lengths, is the product of the odd integers. And so for 21 of them, there are 3 of them for 4 tips, 5, 6, 7, and you go on up, and here's 21. So it's the product of the odd integers up to, I think it's 2n minus 5. And for 21 species, you have this many, which is bigger. That's the point where it gets bigger than Avogadro's number. And so I remember one of the pioneers of molecular evolution, Walter Fitch, when he saw this number, said, wow, we have a gram molecular weight of evolutionary trees. the thing about them is there's a lot of them. They blow up fast. The product of odd integers goes up faster than factorials. And the result is, and what goes with that, although it's not inevitable from that, is that a lot of these methods I'll mention are NP hard, or some of them are not known whether they're NP hard, except that if I were betting, I would bet. I don't think I could get anybody to take the bet, but I would bet in favor that ultimately they will be proven NP hard. I believe there's some proofs now for likelihood that certain cases are NP hard. So this is all NP nasty stuff. And also, I don't think it approximates well either. Now, some of the methods that have been used to infer phylogenies, one of the first ones to use was people to say, well, surely we should arrange the tree so that the minimum amount of change in evolution happened, that we don't have to assume any more change than necessary. And you get a whole class of methods called parsimony methods, and all I've shown you for that here is a simple data set. This is imaginary DNA sequences of 6 sites long and 5 of them. And here's a tree. Now, parsimony methods in the case of DNA infer an unrooted tree, so I should have probably drawn it as unrooted. And what this is the most parsimonious tree. That is, it is the tree that can evolve these sites with the fewest numbers of changes of states, where you assume that at each point along here there was some 6-base sequence, and as it goes along it undergoes single base changes. And here is one particular arrangement, there are more than one, for this tree topology of where the changes are, and there turn out to be 8 of them. And there are good algorithms going back to Walter Fitch's work in 1971 and then generalized shortly after and reaching its greatest generalization, the work of David Sankoff. There are good algorithms for, in a time linear in the number of tips in the tree, working your way down the tree in a dynamic programming algorithm and computing what is the minimum number of changes or the minimum amount of weighted change in more complex cases that you would need for this tree. So any one tree could be evaluated reasonably quickly in basically time linear in, for each site, I should say, it's evaluated separately for each site. And for each site, it's linear in the number of tips. So it's tips times sites is the total effort, although you can economize on that a little bit by identifying multiple sites that have the same pattern of bases and reusing the computation. So it's sublinear in the number of sites, and it's linear in the number of tips. And when you evaluate it, it doesn't really tell you where the changes are. You have to do another pass of going back up the tree, do some further processing to figure out exactly where they might be or what are the different possible places they might be. Now, parsimony methods were very attractive to people. They have some advantages and disadvantages, and I'm going to do that for all of these methods. One of them is the model. It's not model-based. Nobody gave you a probabilistic or statistical model of how these characters arose on the tree. They just sort of arbitrarily decreed this criterion and said, surely that's a good criterion, isn't it? So people using it get the illusion that, oh, I'm model-free. I don't have a model. Isn't it wonderful? Nobody can question my model because I don't have one. And isn't that great? And a lot of the papers I labored on in the 1970s were trying to batter people over the head with the idea that these things had properties and they only behaved well under certain circumstances, and maybe there's really models underlying them, or at least partial models. A point that has not been universally agreed to by everybody. Another advantage is they're reasonably fast. You don't have to mess about with branch lengths on these. Branch lengths don't matter as long as they're not 0. And you can do some trickery as you search different trees for reusing bits of the tree that haven't changed between the two trees you're looking at. And so it's reasonably quick to compute. It's not infinitely fast, but reasonably quick. And you can do your search reasonably quickly. But the problem is still NP hard, and that has to be kept in mind. When amounts of change that you expect on the tree are small, when you're dealing with species that haven't diverged very much, that's one case where they can be shown, parsimony methods can be shown to have good statistical properties. They're very close to what other good methods will do. And they're nice statistically. So they're reasonably fast, and in this case, nice statistically. Disadvantage. When some branches in the tree are longer, particularly when some are long and some are short and they're in the wrong arrangement, when some nearby branches but that are not immediately connected to each other are long, you can also show that there's a phenomenon called long branch attraction that comes in, which has the property that as you accumulate more and more sites, as you make longer and longer, say, DNA sequences, you are more and more certain of putting those two branches connected to each other when in reality they aren't. You get a bias towards the long branches being together on the tree. So it's not just biased, it's inconsistent, statistically inconsistent. It converges on the wrong answer with infinite amounts as you get up to infinite amounts of data. And that's a misbehavior that you have to worry about, and sometimes a serious one. John Hulsenbeck has written papers trying to show that certain cases, I know in insects, that you could show that this was happening. John, you're busy programming, but is that still true? Are the trees of the Strepsiptera really biased? So it's still up in the air, but your conclusion was that there were long branch attraction cases there. Yeah, so there's some real cases. Another disadvantage of it, I think, is it likely to make you think that you've got William of Ockham, philosopher in the 1200s, and the notion of Occam's razor behind you, and that makes people very intoxicated. And I have been the victim of some of that intoxication. And the disadvantage, which really is connected to the very first point, is, but another disadvantage is that since you have some reconstruction of where changes occurred, although it's often somewhat uncertain, it may make you think that you know all that you know that really all those events happened in exactly this particular way. And that's always a mistake to think that, because things are much noisier than that. Another category of methods that was very popular at the beginning, and these methods popped up, I should say, in the 1960s when computers became available. Computers became available to biologists at universities in the late 50s, and by the 60s people started making phylogenies using computers, and parsimony methods came up very early. So did distance matrix methods. And the way they work is you have some data, and we make a table of distances between these. Now, what do we mean by a distance? We mean, in effect, a distance is a two-species tree. There's this sequence and that sequence, and we just put all, what is the tree that is an unrooted tree for two things? It's just a line going between them. And so the only feature it has is a branch length. And you can estimate that by estimating how long would be the branch length that would be needed to achieve the amount of difference that you see.
Hamming distances?
No.
They're not.
They're not. In fact, that's exactly the good point to make right now. The Hamming distance would just be the count of how many symbols they differed by. But in evolution, with any stochastic model of evolution, there will be some cases where A changes to G, and then before it gets to the other end, changes back to A. And so there is a change there that you did. Two changes that you didn't even see. Or an A changes to G and then to C. So what looks like one difference is really has been two or more.
So is it some kind of edit distance?
Edit distance. No, it is not an edit distance. It is that would be a Hamming distance in this case, if you're allowed to replace any letter by any letter. So it's a corrected distance inflated by an amount. You have to use a model of evolution, and under that model you have to make an estimate of what branch length would lead to these two sequences. And in simple cases it would lead to this amount of this size Hamming distance. And so the distance you get is an extra correction on it for this extra unobserved change. And so you have a table showing you the estimates of the length of branches for the two species trees for all pairs. So in effect, it's like we've made a whole bunch of two species trees. Now what we have to do is say, what bigger tree would come closest to fitting those? Well, you take a given bigger tree. Suppose somebody walked in the door and said, I think this is the tree and I think these are the branch lengths. And you now have to evaluate it. What you can do is take this tree and compute the branch length between each pair. And that's what's over here. From A to B is 0.08 plus 0.05 plus 0.10 is 23, and so on. You do that for all of them. And then you say, well, this is the predicted set of two species trees you would get out. This is the set we estimated. Let's make a measure of how close these two distance matrices are from each other. The simplest case, invented back in the 60s, was a simple sum of squares of differences. And there are more elaborate ones involving weighting them so as to de-weight some of the big distances based on the statistical noise involved in those distances.
That's clearly NP. You have polynomial proof, but is it also NP complete?
The finding of this tree, remember what I said was somebody gave you this tree. Now what we actually have to do is go out and wander around somehow in tree space and find a good tree, and for each tree we have to estimate the branch lengths, which can be done by solving a set of normal equations for least squares. That's one way to do it. But we have to do that for each tree topology we look at. And there's some ways of making it a little bit faster. There's some nice papers on speed-ups. But in the end, this also has been proven NP hard. So although I have to say, when Richard Karp used to be at the University of Washington, I would have this terrible problem. I'd get up to orate about what was NP complete and NP hard, and I'd look out in the audience, and Richard Karp is in the audience. And so I would chicken out and say things like NP nasty, because I wasn't absolutely sure that this problem I was talking about was NP hard or NP complete, and there he was. Richard was very nice about it. Okay, so here what you're doing is finding a tree that fits this bunch of two-species trees as well as possible. And this has been a very popular approach, and there are least squares methods, weighted least squares methods, and even generalized least squares methods. And there are methods that simply use clustering algorithms to approximate those. The most popular is a method called neighbor joining that Ney and Saito invented in 1987, and it's very fast. An advantage of these distance methods is they are model-based. You have a probabilistic model, so you know what your assumptions are about what the DNA is doing. Another advantage is its geometry, and because it's geometry, mathematicians fall in love with it. Okay. There are often, it's often fast. Some of the generalized least squares methods are not, but neighbor joining is as I recall, order n cubed in the number of tips in the tree. And it's pretty fast, and you can handle large numbers of sequences. The disadvantage is it is not using the data fully efficiently statistically. It is first reducing this full set of sequences down to a set of pairwise distances and ignoring all other features. And you'd think, well, that'll lose a lot. It'll lose all the triple and quadruple and higher order tuple stuff. But the amazing thing is when you do computer simulations, it's not fully statistically efficient. You can show it's beaten by more efficient methods, but not by much. These things come awfully close. So they're impressive in that respect. That's that point. Now, there are some real other disadvantages, one of which is we have methods, some of the ones I'll show you in a moment, which can accommodate, say, variation of rates along the molecule. And when you use those methods, if you're using a distance method, you can accommodate variation of rates, but not in a way which successfully propagates information about where the fast rates are in the molecule and where the slow rates molecule. If it's comparing, not carrier pigeon, it's dead, but pigeon and penguin, you find that up in the front end of the molecule, sorry, the front end's over here for you. In the front end of the molecule, there's high rates, and in the back end, there's low rates. When you come to look at human and chimpanzee, you've forgotten that, and you're doing it all over again. And you can't propagate that rate except by engaging in some elaborate kludge. So they have some problems using that kind of information, and that may be a serious issue. A disadvantage is that it's geometry. And so once mathematicians become intoxicated by it, you cannot talk them out of using distance methods because it's geometry. Everything has to be geometry, doesn't it? And often you'll hear people who are mathematical say, well, in studying trees, the basic question is how far things are from each other. And the next thing you know, they're off into geometry. But I urge people to be somewhat resistant to thinking, to totally committing to thinking about it as geometry, because it tends to push you into certain methods and away from other methods and in a not necessarily functional way. Now, there came along after these two methods maximum likelihood, which people will often say, I invented. I didn't. Well, R.A. Fisher invented the criterion in 1912 and 1922. I have been at meetings where people said, Joe Felsenstein invented maximum likelihood. And then I feel like, is there a statistician in the room? If so, I want to crawl under a rock, you know. But Anthony Edwards and Luca Cavalli-Sforza in 1964 were the first to apply likelihood to phylogenies for some gene frequency models. And the great statistician Jerzy Neyman was the first one to apply it in 1971 to molecular, this was protein sequence data. In the maximum likelihood methods, you have a tree, you have some branch lengths, and you have a probabilistic model just as you do in the distance matrix methods. And then you're going to compute the probability of the observed data given the tree and the branch lengths. And to do that, if you knew what the base, let's say for DNA, what base existed at each of these four locations, for that assignment, you could simply multiply the probabilities of change in these branches under some simple assumptions of independence of change, that there's a Markov process working up this diagram, and that there are the usual conditional independence assumptions. And when you do that, you get this big product here of probabilities of change dependent on the shape of the tree and the branch lengths. And what you have to do is imagine summing it over all of the assignments of nucleotides to interior branches. That can be done in principle. In practice, there is a dynamic programming algorithm that does that summation without doing the summation, works down the tree, bookkeeping information, and gets you the likelihood by summing all the possibilities. In time, linear in the number of tips, the product of the square of the number of states, which for DNA is 4, and you do it once per site, except again there are some site economies you can do, so it'll be sublinear in the number of, in the length of the molecule. And that gives you, then you have the usual problem of going around all the tree topologies or somehow searching the tree topologies, and for each maximizing the likelihood with respect to the branch lengths. So, advantages. It uses a model, the assumptions are clear. It's fully statistically efficient. It has the usual statistical proofs about asymptotic efficiency. It's computationally slower because you have to optimize with respect to these branch lengths, and you don't have the equivalent of the least squares normal equations that let you do it by matrix operations. So it's slower. It has the advantage of allowing you to use statistical tests by likelihood ratio tests, but you can't use likelihood ratio tests between different tree topologies. I wish you could, but you can't. So that they're not nested models. And the result is that's too bad. But you can do other things. You can test whether a certain parameter in your model is a certain value or not by the usual likelihood ratio test machinery. Wait, was that the last? I think it was for that. Okay. Well, you go beyond likelihood methods, and more recently, Bayesian inference has been very popular. And Bayesian inference, which John Hulsenbeck has been a leader in phylogenies, is basically using the likelihood machinery, all the same models, all the same calculations of the probability of the data given the tree, but adding on prior distributions on the parameters of the model and on the trees themselves. Up until the 1990s, it was not practical to do Bayesian inference on trees because of the computational burden. But then we were able, Markov chain Monte Carlo methods had developed a lot in the 1980s, and by the 1990s they were being applied to phylogenies. And so MCMC does allow us to draw a big sample from the posterior on the trees. I say allow us, it allows, if you use John's program, it allows John to draw a big sample from the posterior on the tree and get a good picture of what the posterior is. It's very popular right now, has risen rapidly in popularity in recent years. One advantage is the interpretation of the result is very straightforward, because what the posterior tells you is what is the probability of different trees given our priors and given our data, given our models. And that's something you don't get out of likelihood. Likelihood tells you what's the probability of the data given the tree, but not the probability of the tree given the data. It's not available to you without the priors. It gives you, in a sense, what you want, the probability of different results. Disadvantage that you always have with MCMC is, well, how long do you have to run? When is enough? And there are various convergence criteria. They're starting with different starting points and seeing if you end up with the same thing. There's a whole bunch of ways, but at least it's something you have to worry about. And there are cases where people come to conclusions, but they haven't run the MCMC long enough, and so their conclusions are, in fact, wrong. Then there's the problem, where do we get priors from, and what effect do they have? And that's always a worry with Bayesian methods. It's the big difference between a Bayesian method, likelihood method, and a Bayesian method is, and you use a prior. So worrying about the prior is really the one additional thing you have to worry about with Bayesian inference. And finally, there is the property of Bayesians who are always going around and telling you that if you'll only sign on to the cause, you'll be much happier. Okay. So now people have been working. I gather that at Microsoft Research, you've done a lot of people. There are people who have done a lot of work on graphical models and on Bayesian inference using graphical models. And so one of the questions you expect to arise in somebody who's been doing that is, we have all this graphical model machinery. Is it possible, shouldn't we think about it as a graphical model? Is it possible to think about these things as graphical models? And if so, couldn't we then make use of all this wonderful work that's been done on graphical models and Bayesian methods and other methods and graphical models? And the short answer is, yes, they're graphical models, but you have to be a little careful. What they look like is this. Here's a tree, nodes with arrows and nodes with arrows. For the site, the first site in the molecule, let's think DNA. But then for the second site, there is another tree that's exactly identical. It's in the simple case, it's a Xerox copy of the first tree. But it's an independent set of events on that tree. So the first site is doing its thing on the first tree, and the second site is doing its thing on the second tree, and so on. But they all share the tree topology and the branch lengths. And you have to imagine this stack of trees going back 500 or so for a sequence of length, say, 500. And on this graphical model, Markov process works, and it has the usual conditional independence assumptions, and it's a graphical model. There's no problem. It very much fits into that. So then you might say, well, you people are about to benefit from all this wonderful work we've done. Well, Molière had a character who was delighted to discover that all his life he had been speaking prose. And we, in the field of phylogenies, discovered that we had been doing graphical models since the 1970s. And then we discovered that all these wonderful graphical model machinery that was out there that we could take advantage of was exactly the stuff we had already been doing. That the dynamic programming algorithm for adding up likelihoods was a central feature of graphical models in a more generalized form, of course. And in fact, we got our machinery from the closely related field of statistical genetics. And we have Elizabeth Thompson here in this meeting to tell us lots about that, and some other people as well. And there they had a bunch of algorithms called peeling algorithms. Ours are, by analogy, called pruning algorithms, which they had developed from about 1970 onwards, which were used for efficient computation of likelihood by dynamic programming methods on pedigrees rather than on branching phylogenies. And we had basically, our machinery is an adaptation of their machinery. And all of us had been speaking prose all this time, that we've been doing graphical models, we just didn't know it. But that's nice, but what it means is no new tools out there as far as I know. Maybe there are some that I don't know about. Now, what about the second major topic? I've taken up lots of time on just how you find good trees, and those are the four major approaches right now. Another one, invariance, you've heard a little bit about from Jun-Young Kim as well. It's less used, but of interest. Well, there are a variety of ways of putting of talking about the statistical support on phylogenies, and one of them is to use bootstrapping, the standard statistical technique of bootstrapping. And bootstrapping, one takes a sample, and it comes from some distribution, and you might, for example, not know that distribution. And you, therefore, can't get more points from that distribution. But what you can do is say, well, maybe, and this was Bradley Efferin's insight, is that maybe this collection of numbers has a distribution that is somewhat similar in shape to the original distribution. And so, if we want to get a sense of how much noise there is in the process, we can resample from our own data sets of the same size. We have to resample with replacement. Now, in the case of phylogenies, in our simplest form of our models, each site evolves independently. There are versions where it doesn't, but let's stick with the simple ones first. So the independent observations are columns of the data matrix, each one a site in the alignment of DNA sequences. Here are the species or sequences down the left side and the sites across the top. So what do we want to do bootstrapping? Well, it turns out the natural way to do that is you sample, randomly draw sites with replacement and start making up a fake data table of the same size. And you do so until you fill it out. There is the possibility that you may draw from the same site twice. That's okay. And when you're finished, you take whatever algorithm you're using, parsimony or distance or likelihood or Bayesian. You're not likely to do this if you're doing Bayesian because you have something else you could do. And you estimate the tree. Now you do it again. You take a new bootstrap sample, number 2, and you estimate the tree. You keep going and say the proper number of samples to take is your year's computer budget divided by the cost of a single replicate. And anyway, you want a large cloud. Actually, 1,000 is usually plenty. 10,000 more than enough. And you get this cloud of trees. And they, I think, are a fair representation of the statistical noise in the inference of the tree. But the question is how to summarize that down to something more compact. And what a lot of people have used, and I advocated, was the use of a majority rule consensus tree, which basically is a tree that has all the branches that occur in more than half of that cloud, more than half of the trees in that cloud. This is a slightly extended version that has a few branches that occur less frequently as well, as long as they're not contradicted by something more frequent. Anyway, what you see is that this is a parsimony analysis of a 14-species DNA sequence data set with 232 nucleotides of mitochondrial DNA. So it's a small data set. And the parsimony run on bootstrap sampled versions of that data set gets this as the majority rule consensus tree. And 80%, we conventionally put two-digit numbers next to each branch to indicate what percentage of the time the branch showed up in the cloud. 80% of the trees had chimp next to human, had this branch here that separates, partitions the data into chimp human versus everybody else. 84%, this one, 84% for this branch had human, chimp, gorilla versus everybody else as a branch in the tree. And you go on down. And what you find here is the great apes are getting about 70 to 80% support for all their branches. Them as a group has 74% support. There are 4 macaques here, old world monkeys. They get very high support. They're there 99% or 100% of the time. The great apes and old world monkeys are a group 77% of the time. That's believed to be correct. The squirrel monkey from South America comes in. It's together with them only 42% of the time. That is believed to be a correct relationship. Then you get out here, and here's a famous primate called the mouse. Well, the mouse has somehow sneaked in here. Down here is the tarsier and lemur, which are primates, prosimians. They're not monkeys, but they're primates. The tarsier is believed not to be here, but branching off the main line. They're not terribly well supported. They're there only 50% of the time. The mouse has sneaked in, and the cow is out there as an out group. They should have both been outside. You can think of that if the cow had sneaked in, this would be the Gary Larson tree where cows are acting like people. If mouse had sneaked in, it would be the Walt Disney tree. This is the Walt Disney tree. Bootstrapping has some alternatives that are similar. One is parametric bootstrapping, which is the same process, but you get your data sets not by subsampling columns, but by using a computer simulation under your model on the best tree you estimate. You computer simulate data sets up that tree. Bayesian inference, when you use Bayesian methods, you get a cloud of trees out naturally as part of your inference. You get a distribution of trees. And that then displaces bootstrapping as and is a more natural assumption in the context of Bayesian methods. And so you get support information, and you can get a probability that chimp and human is a group. It comes out at you from the Bayesian methods. So they're major alternative. And there's another test that I won't go into discussion of, the Kishino-Hasegawa-Templeton test, which can compare pre-specified trees with each other using the individual sites as independent samples and doing paired tests. And there's some multiple tree versions of it, too. Well, that is a very quick skim through how you assess statistical support for different parts of a tree. Let me just talk a little bit at the end about why you'd want to know the tree. The main reason is the order of branching of species affects the history of every gene in the genome. And so studying one region of the genome and inferring the tree gives you information about what to expect in other genes. The information carries over from one part of the genome to another. If your units are species and they share the speciation event, whole genomes go both ways. So it becomes the essential underpinning of comparative biology between species. You have to have a phylogeny and some discussion of the processes of how your characters evolved. The molecular data then illuminates what your measurable characters, your morphology, for example, or behavior are doing. And it all interacts in a very helpful way. An example is that people looking between multiple sequenced genomes to find regions that might be important and might be doing something, the key to which is that they change more slowly than other parts of the genome because they're being held in their sequences are being conserved under natural selection. We can use hidden Markov model machinery, and they're what are called philo-HMMs. And David Hausler's group in the University of California, Santa Cruz has made a lot of use of this, looking for interesting regions of the genome besides the protein coding genes. And philo-HMMs are very big right now. There's another kind of tree. I'll end up just talking about two other kinds of tree that you will see. One of them is called the coalescent, and they're trees of ancestries of copies of a single gene locus. If I take a copy for a protein coding locus like a hemoglobin beta, and I take a hemoglobin beta copy from each of us in this room, and I make a tree, that's not a tree of different species. What it is it's a tree of copies of genes within a species, the same locus. And let me skip to the next slide and show you the picture. If I make a model of reproduction of a population, and I have 10 individuals, and each is diploid, and I go forward generation after generation going upwards. Under the standard models of theoretical population genetics, a field in which I was originally trained in, the Wright-Fisher model, each copy is in effect randomly descended from one of the 2n copies, the 20 copies in the preceding generation. So if you take each copy and draw a random line back to one of the random copies, you get this tangled mess. And if you take a sample out of that population of this copy and this copy and that copy, if you go down, you'll see that its ancestry forms a tree. Here are two lineages coming together in a single point. Finally, it all comes together down here. And there is a tree of ancestry. It is the result of the random events of individual genes getting copied into descendants, not the split of two species from each other. But it's still a tree. You don't have very much power to infer it because the number it's very shallow. This divergence is very recent, and so not many bases have changed. Now let's go back. Each coalescent tree, it turns out, applies to a very short region of the genome, maybe as little as about one gene, because genetic recombination has the property that pieces of DNA in you come from different ancestors. And that means they have different trees. So as you go along the genome, the tree keeps changing, and where humans would have hundreds of thousands of essentially completely different trees from one end of their genome to another. So the coalescent tree for one part of your genome doesn't tell you about everything in the whole genome, just tells you about what happens to that. They do illuminate things, even though they're very hard to infer. They illuminate things about the population. How big it was controls how deep the tree is and how long it takes for lineages to come together. Migration rates, recombination rates, how quickly the tree changes along the genome tells you about that. And you can accumulate information about those parameters across different loci, even though they have different trees. The way to do that is to use Markov chain Monte Carlo methods, and there's a whole wave of development of this by groups in different parts of the world, and Mary Kuhner's group in our lab is doing that and has a large program package for doing that that's some years old. And this is really the interface between within species work in population genetics and between species work on phylogenies. They connect through this work on coalescence. It's also the statistical machinery you need to evaluate and test things about things like the ancestry of humans and mitochondrial Eve, the ancestor of our mitochondria, or Y chromosome genealogies, and also for samples from other parts of the genome, which is an active area right now. One other kind of tree, and then I'll finish up, and then I'll be done, is trees of gene duplications, gene family trees. They are neither a phylogeny nor a coalescent. You have a species, you have a locus, it goes along, the species speciates, you're getting a normal tree here, but here's an event, it's a gene duplication. A hemoglobin locus has duplicated, made a copy in some other part of the genome. Now you have alpha globin and beta globin. And so you have two different genes, and each is going to evolve normally. And they will come to a speciation. Each will go into the descendants quite normally. And so you will get a tree connecting these two different genes. And when you untangle it, here are the yellow dashed lines, the orange dashed lines, are the edges of the species. The solid yellow lines are the descent of the individual gene copies, which is what you see when you drop these sequences into a phylogeny algorithm. If you take the one locus in a frog or the two loci in humans or the two loci in monkey or squirrel and drop them all into a tree algorithm, you get something like this. And what you find is a subtree for the locus A, a subtree for locus B, and they hook together, and that fork is not a speciation, it's a gene duplication event. And then they connect together with the single copy in frogs. So gene duplication trees are another thing, sometimes called gene trees, but that's often also sometimes used for coalescence, so it's a confusing term, and I tend not to use it. So those are three different things called trees. We have trees upon trees upon trees. We have coalescence, gene family trees, and then we have the original the species trees that we've been talking about. So that's all that I'm going to talk about. Here are some books you can look at. This one you buy. The others you think about buying. Semple and Steele is a much more rigorous mathematical treatment. The other day I looked it up on Amazon, and they have a little list of common phrases in the book. And the phrase, one of the most common phrases was proof, let. Mine does not have that. His has that. Okay. Ziheng Yang has just published a very nice book, Computational Molecular Evolution. It's a little misleading. It's really computational molecular phylogenies. He doesn't cover within species evolution. And that's a good survey, and he's been a leading person in this area. So there are these three nice books. If you want to know about programs to do phylogenies, I maintain a website as a hobby almost that started out as a list of 17 programs in my documentation of my own package FileUp, and then it got too big and I had to take it off and it became a website, and now it lists 348 phylogeny programs with more to come. Thank you. Unfortunately,
just
yeah.
wanted to discuss during the coffee break, but
Please.
I have a quick question. When you mentioned likelihood versus Bayesian methods,
Yeah.
you put for Bayesian one of the disadvantages is that where do you get the prior from?
Yeah.
But when you put likelihood, you didn't mention one of the disadvantages where do you get the model of evolution from?
But the model of evolution is shared exactly in Bayesian and likelihood methods, using exactly the same model, so it's a common disadvantage. It's not a disadvantage that's relevant to the comparison between them.
Sure, because it
So.
wasn't listed as a disadvantage for any.
Well, I listed as an advantage for a number of the methods that they had a model. That has the disadvantage that you have to ask, well, how did you get the model? But that's nothing compared to the disadvantage you have if you don't have a model and kid yourself that you're somehow now free of thinking about models. And that's what a lot of the most ardent advocates of parsimony do. Yeah, I'll be around for another hour, and then I have to go to something, but I'll be here tomorrow, and people can catch me anytime.
Thank you very much.